Hello, and welcome to the Bring Your Own Tools video overview for the Seven Bridges Cancer Genomics Cloud, powered by Valsera. In this video, you will learn all about how to create tools or workflows on our platform. For this video, we'll be giving you an overview of the platform and some information about how to bring your own tools. We'll feature two examples, a workflow and an R-Shiny app that have been integrated with the platform. Lastly, we'll share some helpful resources for any questions you may have. The Seven Bridges Cancer Genomics Cloud, powered by Valsera, or CGC for short, is part of a larger ecosystem, the NCI Cancer Research Data Commons, or CRDC. The CRDC is a cloud-based data science infrastructure that provides secure access to a large, comprehensive, and expanding collection of cancer research data. Users can explore and use analytical and visualization tools for data analysis in the cloud. The CGC is a cloud resource highlighted above that was built not only to provide access to CRDC data from the several data nodes, but the analytical tools to analyze it too. The Seven Bridges Cancer Genomics Cloud, powered by Valsera, is a cloud platform that allows you to analyze cancer data where the data lives using the computational speed of the cloud. This means that an analysis that usually takes 1 weekend to complete on your laptop can be completed in 10 minutes running on the cloud. The CGC provides powerful, yet easy-to-use interfaces to empower cancer researchers to draw new insights from the over 3 petabytes of data that are currently being accessed by more than 7,000 users. These users have, in total, built 80,000 workflows and utilized 1,500 years of computation on the CGC. The CGC is a stable, secure, and highly customizable cloud storage and computing platform. And as the CRDC ecosystem has grown, the CGC has grown alongside of it to provide a user-friendly portal to serve researchers, clinicians, data scientists, and more. The CGC serves a wide range of scientists and users with varying skill sets. There are bioinformaticians with some programming experience and developers that are able to create their own tools and workflows. For bench scientists and clinicians that are not as familiar with command line programming, we make it easy to find the tools you need for your analysis without getting bogged down with all the coding. No matter what skill level, there are tools and resources to help. Seven Bridges, which is a part of the Cancer Genomics Cloud ecosystem, provides a secure workspace environment to do data analysis and collaborate. Analyses can be set up using a rich graphical user interface or through scripting via API calls. Private cloud-based JupyterLab and RStudio environments are also available, with Galaxy and SAS Studio coming soon. Cloud compute and cloud storage are available on AWS or Google. There are hundreds of accessible and hosted pipelines to get you started. As mentioned, you can perform custom tertiary analysis and visualizations directly in the cloud using our R and Python tools. Galaxy, SAS, and an imaging service are all coming soon. These work functionally the same way you're familiar with on your computer, but a lot faster on the cloud. Bringing your own tools is simple with our built-in tool editor. This GUI template simplifies the technical aspects of this process and makes it as easy as possible for users to get their tools set up on the platform. The CWL code can also be edited directly in the tool editor if that is desired. For users working with JavaScript dynamic expressions can be tested without ever having to leave the tool editor. Let's take a closer look at how to use the tool editor. In this above figure of our visual editor, the numbered orange icons denote the following. Tabs for tool editor views. Here we have App Info, Visual Editor, and Code. Commands that are available for apps. Shown here, we have revision history, save, run, and additional actions where you can export your app to CWL in JSON or YAML format. A tool editor pane, an object inspector showing properties of an input port of the tool being described in the tool editor. The items shown here vary depending on the object that has been selected, and in some cases on other selections in the object inspector. We have a preview pane showing a preview of the command that is being built up in the tool editor. And lastly, a tool status bar showing the CWL version, and a toggle to show and hide the error state and the command line pane. This GUI template simplifies the technical aspects of building your tool. Once you've built your tool, you can choose to incorporate it into a workflow. String together multiple discrete tools by dragging and connecting them together in our workflow editor. Using this straightforward process, researchers can create workflows that are as simple or as complex as they need for their analysis. One group that has really taken advantage of our workflow and visual editors is Dr. Daoud Meerzaman's lab at the NCI's Center for Biomedical Informatics Information Technology, or CBIT for short. We'll be showcasing some of the work done by this lab. One item is a multi-step workflow called MOPAW that performs gene set analysis based on multiple omics data. The second item is an R-Shiny app called OmicCircos that generates high-quality circular plots for visualizing variations in omics data. First, we'll be discussing MOPAW, which stands for Multi-Omics Pathways Workflow. MOPAW can be described as a point-and-click graphical user interface to multivariate single-sample gene set analysis, or MOGSA, hosted on the Cancer Genomics Cloud by 7 Bridges. This workflow handles data preparation for each given data type, dimensionality reduction, and MOGSA pathway analysis. The omics data include copy number alteration, transcriptomics data, proteomics, and phosphoproteomics data. An optional pipeline add-on allows users to easily download data from TCGA and CPTAC, then pre-process the data to be used for the MOPAW workflow. This workflow and its construction process on the CGC were documented and recently published in the journal Cancer Informatics. In addition to the article, the Meerzaman group created a detailed workflow user guide that is available in the article as well as on the CGC. To implement on the CGC, a Docker image was created which included all required libraries. The Docker image was then pushed to the CGC Docker repository. Apps running the workflow wrapped in common workflow language were then created by pulling the Docker image to the CGC tool visual editor and filling out the GUI template with parameters and scripts. The workflow editor enables users to create full pipelines by linking together multiple discrete tools as shown in the figure. The workflow editor is a drag and drop visual interface that makes it easy to build even the most complex pipelines. The MOPAW workflow, which is depicted in the image, is composed of 3 discrete tools. The workflow currently accepts 3 expression data types given by users to perform 1. Data preparation, 2. Multifactorial analysis, or MFA, and 3. Pathway analysis. Once the workflow is run, publication-ready output images are generated. Here are a few examples. The MOPAW workflow is hosted on the CGC and can be explored by navigating to public projects along the top blue banner, finding the MOPAW card, and then clicking on Copy Project. From here, you'll be taken to a new page with detailed descriptions, example files already preloaded and available to you, and generated outputs that you can visualize and recreate. Our next example is OmicCircos, a simple-to-use app on the CGC for the circular visualization of multidimensional omics data. The primary focus of OmicCircos is to display the genomic data from human and mouse with built-in genomic coordinates. The data can be gene or chromosome position-based values. These values can come from mutation, copy number, expression, and methylation analyses. The package can easily be applied to any other genomes with customized genomic information. In fact, any data that meets the minimal requirements of positions and relationships can be used. The OmicCircos package can display variations in scatter plots, lines, and text labels. The relationships between genomic features can be presented in the forms of polygons and curves. By utilizing the statistical and graphical functions in the R/bioconductor environment, OmicCircos is able to draw box plots, histograms, and heat maps from multiple sample data. Each track is drawn independently and interactively, which allows the user to optimize the track quickly and easily. Here's another example plot generated with OmicCircos, and an explanation of how to read it. The circular tracks from the outside to inside are as follows. The outermost band represents genome positions by chromosomes. The black lines here are cytobands. The next track is an expression heat map. In this example, red represents upregulated expression and blue is downregulated. The third track is copy number variations. Red here represents gain and blue is loss. Lastly, the innermost track in the plot represents correlation p-values between expression and copy number variance. OmicCircos can be found by navigating to the public apps along the top blue banner of the CGC, then selecting Interactive Web Apps. You'll be taken to a new page where you can select OmicCircos. Finally, you'll be on the OmicCircos splash page. This page features a user guide and references to publications that utilize OmicCircos to plot data. When you're ready to create your own plots, click on Start Discovery. Using the interactive app is as simple as selecting the plot you'd like to create and loading the data you'd like to use, either your own private data or included data. A new plot will be added, and you can click to generate the plot and visualize it. Here are just a few examples of plots generated by other research labs on a wide variety of research topics. These can be found by clicking References on the OmicCircos splash page. If you're ever feeling unsure of where to start or what your next steps are, please don't hesitate to contact us. You can find a little blue question mark button on any page of the CGC platform. Clicking this will connect you with our 24/7 support team. We also have office hours twice a week. This is an informal meeting space where users come to ask questions, share success stories, and more. Join us by scanning the QR code or visiting cancergenomicscloud.org for a link. Thank you for your time.